This is Chapter 2. We're going to be looking at the chemical level of organization. This isn't a chemistry class, so a lot of these chemical concepts we're going to go over very briefly. We're really going to be just looking very briefly at chemical bonds, how chemical reactions occur. So don't worry if chemistry isn't your thing. We're going to be breezing through a lot of this. We mostly are just going over this chapter just so we can get a basic understanding of some chemistry concepts so that we can understand physiology of some of the systems when we look at it in the later chapters. A couple of basic principles of chemistry. Chemistry is just the science of structures and interactions of matter. When we're talking about matter, this is anything that has mass and takes up space. So mass is the amount of matter that a substance contains, whereas weight, on the other hand, is the force of gravity acting upon that mass. Now, matter exists in three forms: solid, liquid, and gas. Now the difference between these is the distance between each molecule and the amount of energy or movement of each molecule. So in solids, the molecules are really close together and have very minimal movement, whereas in a gas, each individual molecule is very far apart, relatively speaking, and they have a lot more movement. All forms of matter, living and non-living, are made up of a limited number of building blocks that are called chemical elements. Each named element is designated by a chemical symbol. A chemical symbol is usually one or two letters of the element's name in English or in Latin usually. 26 different chemical elements normally are present in your body. Just 4 of these elements, though, are called the major elements, and they constitute about 96% of the body's mass. And these elements are oxygen, carbon, hydrogen, and nitrogen. Each element is made up of atoms, the smallest units of matter that retain the properties and characteristics of the element. Atoms are made up of protons, which are positively charged particles, neutrons, which are neutral particles, and electrons, which are negatively charged particles. Atoms have a dense central core, which is called the nucleus. And this consists of protons and neutrons, and then the electrons will move about within certain regions around the nucleus in electron shells. Now remember, you don't have to memorize any of these small specific details of chemistry. We're just going to be looking at these just to get a broad concept of chemistry that we're going to be using in a little bit more detail down the road in different later chapters. Okay, the number of protons in a nucleus is an atom's atomic number. And atoms of different elements have different atomic numbers because they have different numbers of protons. The mass number of an atom, this is going to be the sum of its protons and neutrons. Carbon here, for example, has 6 protons and 6 neutrons. And so its mass number is 12. Even though atoms of one element have the same number of protons, they may have different numbers of neutrons, and so they have different mass numbers. For example, this carbon could be 12 or 13 mass number, so it could have 6 or 7 neutrons in the nucleus. Atoms of the same element have the same number of protons, but they do have ways of losing, gaining, or sharing their electrons when interacting with other atoms to achieve stability. If an atom either gives up or gains electrons, it becomes an ion. Now we'll definitely look at ions in later chapters, so this is going to be a term that you should know. An ion is an atom that has a positive or a negative charge because it has unequal numbers of protons and electrons. So if it had equal numbers of protons and electrons, the positive charge from the protons and the negative charge from the electrons would cancel each other out to be a neutrally charged atom. Whereas an ion, on the other hand, has an unequal number of protons and electrons. For example, if it has 5 protons or 5 positive charges and 6 electrons or 6 negative charges, the overall charge of the atom would be -1. When 2 or more atoms share electrons, the resulting combination is called a molecule. Now, a molecule may consist of 2 atoms of the same kind, like an oxygen molecule, which is going to be O2. So that's just going to be 2 oxygen molecules bound together to make a molecule. Or a molecule may be made of 2 or more different atoms, like water, which is 2 hydrogen atoms and 1 oxygen, so H2O. Then a compound. This is a substance that contains atoms of 2 or more different elements. Most atoms in the body are going to be joined into compounds. So a compound, for example, H2O is a molecule, but it's also a compound. Sodium chloride, NaCl, is also a compound because it's a substance that contains atoms of 2 or more different elements. So a molecule of oxygen, for example, this O2, is not a compound because it consists of atoms of only one element. It's just made up of oxygen. So O2 is a molecule but not a compound, whereas H2O would be a molecule and a compound. Now, the forces that hold together the atoms of a molecule or a compound are called chemical bonds. Now, the likelihood that an atom will form a chemical bond with another atom really depends on the number of electrons in its outermost shell, which is called its valence shell. So the electrons in this shell, this outermost shell, really determine if it can bond to another atom. So here we can see how different atoms will bond to each other. So an atom with 8 electrons in their valence shell, remember their outermost shell, is going to be chemically stable. So valence shell with 8 electrons is stable. And when it is stable, it means that it's unlikely to form chemical bonds with other atoms. So the atoms of most biologically important elements do not have 8 electrons in their valence shells, which means that in the right conditions, 2 or more atoms can interact in ways that produce a chemically stable arrangement of 8 valence electrons for them. So this is what chemical reactions aim to do. Chemical reactions aim to create valence shells that have 8 electrons in them if they don't already. When atoms lose or gain 1 or more valence electrons, this is when ions are formed. So positively and negatively charged ions are attracted to one another. So this is the opposites attract. The force of attraction that holds together ions with opposite charges is called an ionic bond. So let's consider sodium and chlorine atoms as we have here. So this green atom is a sodium atom, and here we have a chlorine atom. So these are going to be the components of table salt. So here, sodium at the top, we can see that it has 1 valence electron. So in this outermost shell, there's 1 electron, but in the rest of this outermost shell, there's nothing. So it's just going to be just this 1 electron is in the valence shell. So because it only has 1 electron and not 8, this is an unstable atom here. So it wants to have a reaction in order to get 8 electrons in its valence shell. If it loses this electron, it's left with 8 electrons in its valence shell. So what happens is that it loses it, then the next shell below this one becomes the new valence shell, because now it's the new outermost shell. So if it loses one, now it has 8 in its new valence shell. As a result, the total number of protons exceeds the number of electrons. So then sodium becomes a cation. A cation is a positively charged ion. Positive ion. So we write cation with a positive plus sign like this, so that you know it's a cation. In contrast, chlorine on the bottom here, this yellow atom, has 7 electrons in its valence shell. So we can see here, it's a valence shell, 1, 2, 3, 4, 5, 6, 7. So if it gains an electron from its neighboring atoms, it will have a complete octet, or 8 electrons in its valence shell. So sodium is going to donate its electron, and chlorine is going to accept the electron. So after gaining the electrons, or gaining this 1 electron rather, the number of protons is going to be less than the number of electrons. So that means that chlorine becomes an anion, or a negatively charged ion. Anion is going to be a negatively charged ion. And we can see that with this here. When an atom of sodium donates an electron to chlorine, the resulting positive and negative charges are going to pull these ions close together, forming an ionic bond. Now, another type of bond is called a covalent bond. Now, when a covalent bond forms, 2 or more atoms share electrons rather than gaining or losing them like we saw in the last slide. Now, a covalent bond may form between atoms of the same element or different elements. These types of bonds are the most common chemical bonds in the body. So we can have a couple different types of bonds. So we can have a single covalent bond, and this forms when 2 atoms share an electron pair, like we see here with hydrogen. So at the top here, we have each hydrogen atom has a single electron that they want to share with each other. So they share them between each other, creating this single bond. So a single line drawn indicates that there's 1 pair of electrons between them, or shared between them. A double covalent bond is when 2 atoms share 2 electron pairs, or 4 electrons, like we see here with oxygen. So each of these oxygens have 2 electrons that they want to share together, so there's 4 electrons between them shared, and it's shown as a double line. So that's a double covalent bond. And then a triple covalent bond is when 2 atoms share 3 electron pairs, or 6 electrons. Again, so we can see here 3 electrons and 3 electrons. Shared between them is going to be 6, denoted with 3 lines, or a nitrogen molecule. Water molecules form polar covalent hydrogen bonds with each other. So a hydrogen bond forms when a hydrogen atom, shown here in gray, and it has a partial positive charge. It's denoted by this little symbol here. And this attracts the partial negative charge of the neighboring electronegative atoms. So this is going to be an oxygen with a partial negative charge here. So electronegativity This is the strength of an element to attract electrons towards itself. So when we have water, so we have an oxygen molecule which is bonded to a hydrogen molecule here and here. So they each have a single bond between them, which means that they share 2 electrons between each of them, making a bond. But oxygen is more electronegative, which just means that it's going to be stronger and pull electrons closer to itself than hydrogen. So instead of these electrons being directly in the center, these electrons are actually going to be held closer to oxygen because oxygen is just going to be a much stronger atom, and it's going to pull those negative electrons closer to itself than hydrogen, because hydrogen is not very strong and it can't pull those electrons very close to itself. But oxygen is very strong. It pulls those negative electrons closer to itself. It is more electronegative. So because these electrons are much closer to oxygen, it kind of gives oxygen a little bit of a negative charge because those electrons are negatively charged. And because they're a little bit closer to oxygen, the hydrogen atoms will each be a little bit positive. Now they don't have a full positive charge because they didn't completely lose those electrons, but they're just closer to oxygen, so we'll just give them a partial positive. So this partial positive will be attracted to a partial negative charge, and that's how we get these hydrogen bonds. Now these hydrogen bonds that form between water molecules, they give water considerable cohesion. Now cohesion is the tendency for like molecules to stay together. Like molecules So this increased cohesion allows water to have a really high surface tension. So water has a high and surface tension is the measure of difficulty of stretching or breaking the surface of a liquid. So if it's a high surface tension, it's very difficult to break the surface of the liquid. And we can see this a lot with bugs that skate along water. They don't just fall directly through the water. It actually the water holds together so well that if a bug is really light, they can essentially just skate along the water. And that's because these hydrogen bonds allow water to have very high cohesion and gives the surface a very high surface tension because those water molecules just stick together so well. Okay, just briefly looking at chemical reactions. So chemical reactions occur when new bonds are formed or old bonds are broken. We always have the starting substances are called reactants. They're usually on the left side of a chemical equation. And the resulting substances are called products. They're usually on the right side. So here we can see that we have actually more bonds, but here we have one bond broken, this bond is broken, and these 2 bonds are broken. And then we have 4 new bonds are created. And this is converting an oxygen and 2 hydrogen molecules into 2 molecules of water. Now 2 major forms of energy are potential energy and kinetic energy. Potential energy, this is going to be the amount of energy stored in a substance due to position. So this is positional energy. So objects really high off the ground have a very high potential energy. Kinetic energy, on the other hand, this is the energy associated with matter in motion. So it's motion energy. Chemical energy is another type of energy. It's actually a type of potential energy. So it's a potential energy, and chemical energy is a type of potential energy that is stored within the bonds of compounds and molecules. Now, the law of conservation of energy states that energy in the universe is constant, and that energy cannot be created nor destroyed. It can only be converted. So this is going to be very important. One other thing that I will mention here is that chemical energy states that when we break bonds, break these chemical bonds, energy is usually released. And in order to create new bonds, we need to input energy. So this is a little energy diagram, and this is how we transfer energy in the form of chemical energy. So a couple different types of reactions here, exergonic versus endergonic reactions. So in exergonic reaction, this is where energy is lost or the reaction releases energy during the reaction. So this is energy is released. In an endergonic reaction, this is where energy is put into the reaction or energy is absorbed. So here on the right we have a energy transfer diagram. So on the left here we have the potential energy. We can also call this the chemical energy. And then on the x-axis is just going to be the progress of the reaction, or just time. So here we have energy of the reactants. It's going to be this line here. And then we have this term come up, activation energy. So this is the minimum energy required for a reaction to progress. So essentially, this is going to be the energy required to break the bonds of the reactants. Break the bonds of the reactants. So kind of have to get over this energy hump and then have this big drop in energy to get to down here, which is the energy of the products. So we can kind of see these lines here. This is the energy absorbed to start the reaction, to break those bonds of the reactants. And then here is the energy released as new bonds form. So we kind of start up here and we end about down here. So this is the amount of energy that's released. So because this is energy released, this is an exergonic reaction. Catalysts. These are chemical compounds that speed up chemical reactions by lowering the activation energy required by a reaction. So here in the red, for the red line, we have the activation energy needed without a catalyst. So this is just the normal amount of energy required to break those bonds of the reactants. But with a catalyst, you can see this activation energy drops substantially. So this amount of energy we need is so much lower. Now, catalysts are used in reactions, but they're not used up in reactions so that they can be continually used later. Just a very brief thing on organic versus inorganic. So organic usually means carbon containing. So organic compounds usually have carbon. They always contain hydrogen as well, and they always have covalent bonds. Inorganic compounds are going to not have any carbon molecules, and usually they're quite simple. Water is really the most important and abundant inorganic compound in all living systems. And nearly all of the body's chemical reactions occur in a watery medium. Now, water has many really important properties that make it a really important compound and a required compound for life. Now, one of the most important properties water has is its polarity. Now, polarity is that uneven sharing of electrons, which puts a partial negative charge on the oxygen and two partial positive charges on each hydrogen atom. So polarity, this is going to be unequal sharing of electrons. So we can see that here with that partial positives on the hydrogen and the partial negative on the oxygen. Again, that's because those electrons are going to be pulled closer to oxygen than to the hydrogen atoms. This property of water makes it a really excellent solvent for either ionic or other polar substances. This property also gives water molecules really good cohesion. Remember, cohesion is the tendency for water molecules to stick together, and it allows water to resist temperature changes. So being polar makes it an excellent solvent. So solvent is a medium for things to dissolve in. So solvent for ionic or polar substances. And that's because we'll find out later that like dissolves like. So polar substances can dissolve other polar substances, but polar substances cannot dissolve non-polar substances. And we'll go into that a little bit later too. Water as a polar substance also gives water molecules good cohesion. Remember, that gives it a high surface tension. And then we're also going to allow water to resist temperature changes because of this polarity. So water as a solvent in a solution, the solvent dissolves another substance called the solute. So solvent does the dissolving. And the solute is the dissolved. Now, the versatility of water as a solvent for ionized and polar substances is due to its polar covalent bonds and also its bent shape. And this allows each water molecule to interact with several neighboring ions or molecules. Solutes that are charged or contain polar covalent bonds are hydrophilic. Hydrophilic. So these are going to be charged ions or polar covalent bonds. And hydrophilic literally just means water loving. Hydrophilic substances dissolve really easily in water. So common examples of hydrophilic solutes are going to be sugar and salt. So here we have salt. We have crystal of sodium chloride. So we can see that the sodium ion can be dissolved by having these partial negative oxygen atoms completely surround each positive ion of sodium. And the same happens with chloride, where those partial positive hydrogens are all going to surround that negative chloride ion. So this is what dissolving is, that each ion is going to be completely surrounded by molecules of the solvent, in this case, water. Molecules that contain mainly nonpolar covalent bonds, these are going to be hydrophobic. So this is going to be water fearing. So these are going to be not very soluble at all. So these are going to be nonpolar covalent bonds, or they're going to be made up of nonpolar covalent bonds. Water fearing. So these are going to be things like animal fats, vegetable oils. We know that oils don't really dissolve very well in water, and this is because they are hydrophobic. Now, water is really such a great medium. We use it for a lot of processes. For example, a hydrolysis reaction. So in a hydrolysis reaction, water is going to be added in order to break bonds. Go in the other direction, we have dehydration synthesis. This reaction, this is where water is removed in order to make bonds. So here, this reaction on the bottom here, we have glucose and fructose on the left and sucrose on the right. Now, when we go from the left 2 molecules to the right 1 molecule, this is a dehydration synthesis. This is where we take one OH molecule from glucose and one H atom from fructose. Those are going to be removed, producing water, and we're going to get a bond between this oxygen right here and the carbon molecule right here. So we get a bond kind of right between these 2 atoms here, and it looks like this. So removing that water molecule, 2 hydrogen atoms and an oxygen atom, produces water and allows that bond to be formed between glucose and fructose. Go in the opposite direction, hydrolysis reaction. This is where we are adding water to break this bond. So we're going to be breaking this bond here by adding water. Now, the specifics of how this reaction occurs is not really important. Really, the actual reactions themselves are not really important for this class either, but just understanding that water is really important as a medium, and it can be used to break bonds, as well as removing water creates bonds. So we're going to briefly go over a couple of these properties of water that make it a really good medium. First, we're going to go over its thermal properties. So one is that water has a really high heat capacity. Now, heat capacity is the number of heat units that are required to raise the temperature of a substance by 1 degree Celsius. So heat and temperature are not the same, and having a high heat capacity means that water can essentially absorb a lot of heat without changing its own temperature too much. And a real-world example of this would be in areas near the ocean. They're quite temperate regions, which means that they don't really get extreme temperatures. And this is partly because of water's high heat capacity. So if the air in the area is very hot, then the water in the ocean will essentially absorb a lot of that heat without changing its temperature too much. So water actually has to absorb a large amount of heat in order to increase its temperature by even 1 degree Celsius. We don't see the water in the ocean boiling because it absorbs a lot of surrounding heat. The next thermal property of water we're going to briefly discuss is the high heat of vaporization. Now, heat of vaporization is going to be the amount of heat required for a solution to change phases from a liquid to a gas phase. So the amount of heat that's required to input into a substance in order to change phases, in order to vaporize from a liquid into a gas. Now, compared to other substances, water can absorb and release relatively large amounts of heat with only a very small change in its own temperature. So, like I said, this is why you have really temperate regions by the ocean, where the temperature doesn't have really high extremes like temperature that we see inland, like where we are. Here we have very high extremes because we're not close to a large body of water. So water not only can absorb a lot of heat without a lot of change in its own temperature, but it also releases a lot of heat. Now, water is also a very good lubricant. It's a major component of mucus and other lubricating fluids throughout the body. And lubrication is really especially important and necessary in the chest for our pleural and pericardial cavities. Also in the abdomen, in our peritoneal cavity, and really we need lubrication where our internal organs touch and slide over each other. So water really helps in protecting your organs. Okay, so a mixture, this is a term we should know. This is a combination of elements or compounds that are physically blended together, but they're not bound together by chemical bonds. So there's 3 types of mixtures. First one is a solution. So solution, this is I'll put this up here. This is a liquid mixture. These are all going to be liquid mixtures though. A liquid mixture in where the minor component, which remember the minor component is going to be the solute. The solute is going to be uniformly distributed within the major component, the solvent. So the solute is the minor component, solvent, ENT, going to be the major component. So solution, this is where solute is uniformly distributed within the solvent. So this can essentially be thought of like salt water, where the salt is the solute and the water is the solvent in which the salt is dissolved in. Next we have a colloid mixture. This really just differs from a solution in the size of the particles. So a colloid solution is going to have, or colloid mixture rather, is going to have larger particles of solute than a solution. So these particles are going to be much larger, and because of this, they usually scatter light. So these solutions are often translucent or opaque. So you can't see very well through them. Last is a suspension mixture. So a suspension is a solution in which the material may mix with the liquid or suspending medium for a time, but eventually it's going to settle out. So the solute is going to settle. I'll put this here. Solute settles. So that it doesn't stay mixed within the solvent. So blood is going to be a really good example of a suspension where the blood cells and other large particles stay suspended in the blood for a time, but eventually if you leave it sitting for a bit, it eventually will settle out and all kind of settle to the bottom. When inorganic acids, bases, or salts dissociate, so this is a term we should know also, so dissociate means that they separate into ions. So into ions. When these separate, they become surrounded by water molecules. Very similar to a couple slides ago where we saw salt that becomes, that dissociates and then becomes surrounded by water molecules, kind of in a solution. Now in acid, this is going to be a substance that dissociates into one or more hydrogen ions, and then also one or more anions. So remember, those are negative ions. A base, by contrast, dissociates into one or more hydroxide ions. Hydroxide ions, remember, are OH negative. And then it will also dissociate into one or more cations. Remember, cations are going to be those positive ions, positively charged. A salt, when dissolved in water, dissociates into cations and anions, neither of which is going to be hydrogen or hydroxide. In the body, salts like potassium chloride, like we see here, are electrolytes. And these electrolytes are really important for carrying electrical currents. So electrical currents are really just ions flowing from one place to another. And electrolytes in the body are especially really important in nerve and muscular tissues. Now, to ensure homeostasis, the intracellular and extracellular fluids must contain almost balanced quantities of acids and bases. So the more hydrogen ions that are dissolved in a solution, the more acidic that solution is. And the more hydroxide ions, the more basic or alkaline the solution is. So an increase in hydrogen ions is an increase in acidity. And an increase in hydroxide ions is going to be an increase in alkalinity. Or basic. So a solution's acidity or alkalinity is going to be expressed on a pH scale like we see here. And this pH scale is going to extend from 0 all the way to 14. Now really this scale is based on the concentration of hydrogen ions in moles per liter. You don't have to memorize the amount of hydrogen ions in moles per liter per pH or anything like that, but this is just for your information. Now the midpoint of the scale here is going to be a pH of 7, and it's at this point where the concentrations of hydrogen ions and hydroxide ions are going to be equal to each other. So this is going to be like in pure water, which is neutral. Now a solution that has more hydrogen ions than hydroxide ions is going to be an acidic solution and has a pH below 7. So pretty much anywhere down here is going to be an acidic solution. And we can kind of draw a line here So kind of right here, if we draw a line approximately across here, maybe a little bit higher, but approximately there, this is going to be our neutral. Not a great straight line, but that's okay. So anything right here at 7. So pretty much anything here is going to have a higher concentration of hydrogen ions than hydroxide ions, as we can see here. And anything to the right of this line is going to have more hydroxide ions than hydrogen ions. So a solution that has more of these hydroxide ions than hydrogen ions is going to be basic and has a pH above 7. So pretty much above 7 to 14 is going to be basic or alkaline, and a pH of 0 to 7 or just below 7 is going to be acidic. Okay, let's briefly talk about buffers. So buffers function to convert strong acids or bases into weak acids or bases. Now, because strong acids and bases can change pH really drastically and quickly, they can really disrupt the body's metabolism. So weak acids and bases, they don't ionize very easily, they don't dissociate very easily, and because of this, they have much less of an effect on pH. Strong acids and bases, they dissociate very quickly and they release their hydrogen ions or hydroxide ions into solution, and this changes pH very quickly. So buffers essentially work by removing or adding protons into the solution in order to keep the pH steady. Now, carbon has a lot of properties that make it really particularly useful to living organisms. It can form bonds with 1 to thousands of other carbon atoms to produce really large molecules that have a variety of different shapes. Now, because of this, the body can build many different organic compounds with carbon. Remember, organic means carbon containing. Now, the large size of most carbon-containing molecules and the fact that some do not dissolve easily in water, this makes them really useful materials for building body structures. Remember that carbon-containing molecules, or a lot of carbon-containing molecules, are going to be nonpolar. Not all, but a lot of carbon-containing molecules are nonpolar. And so this means that they do not dissolve easily in water. Now, organic compounds are usually held together by covalent bonds. And remember that this means that they share electrons. Now, really briefly, we're going to go over some carbohydrates. These are going to include sugars, glycogen, starches, and cellulose. In humans and animals, carbohydrates function mainly as a source of chemical energy for generating ATP needed to drive metabolic reactions. Of all of the types of carbohydrates, monosaccharides, these are going to be the most soluble in water, and this is because they have a higher ratio of polar bonds to nonpolar bonds. There's going to be 3 major types of carbohydrate groups. They're really just different sizes of the same base compound. Monosaccharides, disaccharides, and polysaccharides. Monosaccharides are going to be simple sugars, so just like one single carbohydrate or one single saccharide, monosaccharide. Disaccharides is going to be a combination of 2 monosaccharides, and polysaccharides is a combination of 3 to hundreds of monosaccharides. Okay, so here we have monosaccharides. These are the monomers of carbohydrates. They contain 3 to 7 carbon atoms, And when looking at these molecules, when we look at this type of drawing, there's going to be a carbon molecule, or carbon atom rather, at each junction here. So there's carbon here, carbon here, carbon here, and carbon here. So when we look at these types of drawings, whenever we have kind of an angle in the molecule, this is where there is a carbon. They just don't write in the C. In this diagram, we do have some examples of a few pentoses in the top. This means that they contain 5 carbons. And we do have some hexoses as well. These mean that they contain 6 carbon atoms. Cells throughout the body will break down glucose, is going to be the main monosaccharide that we use to produce ATP. The rest are used in lower amounts. Deoxyribose and ribose are going to be monosaccharides that we see in DNA and RNA. A disaccharide is a molecule formed from a combination of 2 monosaccharides. Remember that dehydration synthesis puts 2 monosaccharides together into a disaccharide. We just saw that very briefly earlier, but we can see that now they have this bond between them. So remember that dehydration synthesis is where water is removed to form these larger molecules. So for example, we have 2 glucose molecules are bound together to form maltose. And another example would be galactose and glucose are formed. They bind together to form lactose. A third major group of carbohydrates is polysaccharides. So each of these polysaccharide molecules contains tens to even hundreds of monosaccharides. Again, these are going to be joined together through those dehydration synthesis reactions. Unlike the simple sugars and the monosaccharides that we talked about, polysaccharides are usually really insoluble in water, and they don't taste sweet like those simple sugars that we discussed. The main polysaccharide in the human body is glycogen, like we see here. Glycogen is made entirely out of glucose monomers that are linked together in these branching chains. So this is how glucose is stored in the body. We're going to see this a lot more later on when we're looking at liver and skeletal muscle storage of glucose. This glycogen molecule, I guess it's not labeled here, but this is a glycogen, this large molecule, and this is how glucose is stored in the body. Okay, just a little bit about lipids. This is a second really important group of organic compounds. And like carbohydrates, lipids contain carbon and hydrogen and oxygen. Now, unlike carbohydrates, lipids do not have a 2-to-1 ratio of hydrogen to oxygen, so there's going to be fewer polar covalent bonds. And as a result of this, most lipids are really insoluble in polar solvents like water. So this makes them very hydrophobic. Or water hating, so they don't dissolve in water. Now, among the simplest lipids are the fatty acids. And the fatty acids are used to synthesize triglycerides and phospholipids. Phospholipids are used in your cell membranes. Now, fatty acids can also be catabolized or broken down to generate adenosine triphosphate, also known as ATP. Fatty acids consist of a carboxyl group. We can see that right here. Have that carboxyl group, that OH, and also a hydrocarbon chain, which is going to be this part of the fatty acid molecule, hydrocarbon chain. Now, fatty acids can either be saturated or unsaturated. So saturated or unsaturated. And a saturated fatty acid kind of like we see at the top here, this contains only one bond between each carbon atom and hydrogen atom in the hydrocarbon chain. So these are going to be single covalent bonds. You can see each of these single covalent bonds. Now, because they lack double bonds, each carbon atom in the hydrocarbon chain is going to be essentially saturated or full up with these hydrogen atoms. Now, an unsaturated fatty acid contains one or more of these double covalent bonds between the carbon atoms in the hydrocarbon chain. So the fatty acid is not completely saturated with hydrogen atoms. So we can kind of see this down here, this unsaturated fatty acid here. So this unsaturated fatty acid has a kink or a bend at the site of the double bond. So here we have this bend in the fatty acid. So if there's only one double bond in the hydrocarbon chain, this is going to be monounsaturated, and it has just one bend in it. The most plentiful lipids in your body and in your diet are going to be triglycerides. So a triglyceride consists of two types of building blocks. A single glycerol molecule, which is going to be this in the light blue, and three fatty acid molecules. So 1, 2, and 3. Well, that's a 3 there. So a 3-carbon glycerol, so we have 1, 2, 3. This forms the backbone of a triglyceride. And then the 3 fatty acids are going to be attached by a dehydration synthesis reaction, 1 to each carbon of the glycerol backbone molecule here. Now, triglycerides are going to be stored and deposited in adipose tissue or fatty tissue, and they provide fat for insulation and protection in the body. So with little to no fat, your joints will grind and bone movement is going to be really difficult, but really too much isn't good either. So just kind of the right amount of triglycerides is important. Now, triglycerides are also the body's most highly concentrated form of chemical energy. So they provide more than twice as much energy per gram as carbohydrates and proteins. So they are an important source of energy. Then we have phospholipids. So just like triglycerides, phospholipids have a glycerol backbone. So here we can see this glycerol backbone. And phospholipids will have 2 fatty acid chains attached to the first 2 carbons of the glycerol. So the top portion of the molecule, which is called the head, this is very polar and can form hydrogen bonds with water molecules. The 2 fatty acid tails, by contrast, are very nonpolar, and they can interact with other lipids or other nonpolar substances. Now, molecules that have both a polar and nonpolar section are said to be amphipathic. So this means that they have a polar section and a nonpolar section. And this is within the same molecule. Now, the next molecule we're going to briefly discuss are steroids. And the structure of steroids differs quite considerably from that of triglycerides. Steroids have 4 rings of carbon, as we can see, that are colored in gold here. And almost all of the steroids that are in your body are going to be derived from cholesterol. They have a polar hydroxyl group that makes sterols weakly amphipathic. So remember, amphipathic compounds are going to be compounds having both hydrophobic and hydrophilic parts. So steroids are going to be mostly hydrophobic, but this little hydroxyl group makes it very weakly amphipathic. Cholesterol is used for cell membrane structures. And then things like estrogens and testosterone, these are used for regulating sexual functions. And then cortisol. This is going to be a really important steroid necessary for maintaining normal blood sugar levels. Next, we have proteins. Proteins, these are going to be very large molecules that contain carbon, hydrogen, oxygen, and even nitrogen. There are some proteins that also contain sulfur. These are going to be much more complex in structure than carbohydrates or lipids. And proteins have many roles in the body. They're going to be largely responsible for the structure of body tissues. So they're going to be very, very structural compounds. There are many different types of proteins, one of these being enzymes. Enzymes are proteins that speed up most biochemical reactions. There are other proteins that work kind of like motors to drive muscle contraction. There's also going to be other proteins called antibodies that defend against invading microbes. So lots of different types of proteins, and we'll talk about a lot of them in more detail a little bit later on in the class. The monomers of proteins are amino acids. So amino acids, these are going to be the smallest building blocks of the proteins in your body. Now there are 20 different amino acids that combine together to produce different proteins. And each amino acid is made up of a hydrogen atom here and 3 important functional groups that are attached to a central carbon. So there's going to be an amino group on one side, a carboxyl group on the other side, and then a side chain on the 4th side. The amino group, carboxyl group, and hydrogen atom are going to be the same for every amino acid. It's just going to be the side chain that changes for each amino acid and is the variable region. A protein is going to be synthesized or created in a stepwise fashion. So what this means is that one amino acid joins the second, and then a third amino acid comes and joins the initial 2, and so on and so forth. So that one amino acid is added at a time. The covalent bond joining each pair of amino acids is called a peptide bond. It has its own little special name. Now, as the peptide bond is formed, a molecule of water is removed, so this makes this type of reaction a dehydration synthesis reaction, like we've seen before. Now, when two amino acids combine, a dipeptide results. Adding another amino acid to a dipeptide produces a tripeptide. And then further additions of amino acids results in the formation of a very chain-like peptide that we usually just call a polypeptide. Proteins are polypeptides, and proteins exhibit 4 levels of structural organization. So our first level here is called a primary structure. It's going to be our furthest on the left here. A primary structure. And this is just the unique sequence of amino acids that are linked by these covalent peptide bonds, forming this polypeptide chain. So this is strictly just the sequence of amino acids, the order of the amino acids that make up the proteins. This is just the primary structure. Then we have the secondary structure. So the secondary structure of a protein is the repeated twisting or folding of neighboring amino acids in the polypeptide. So the primary structure will twist and fold, making up a secondary structure. Then we have a tertiary structure. This refers to the three-dimensional shape of a polypeptide chain. So each protein has a very unique tertiary structure that determines how it'll function. So our functionality comes from the shape of the tertiary structure. Some proteins stop here. They just have a tertiary structure. They're made up of a single chain of amino acids that are folded and twisted into this kind of a glob structure. There are other proteins that contain more than one polypeptide chain. And so this is where we get a quaternary structure. This is the arrangement of individual polypeptide chains relative to each other. So we kind of have this blue polypeptide and a gray polypeptide in this quaternary structure here. Now the bonds that hold polypeptide chains together in a quaternary structure are quite similar to those that maintain the tertiary structure. So we're going to get bonds that kind of hold it together in this three-dimensional shape. In living cells, most catalysts are protein molecules that we call enzymes. Now, enzymes catalyze very specific reactions, and they do so with really great efficiency and with a lot of built-in controls. Now, there are 3 important properties of enzymes. The first one is that they are very highly specific. So I'll number this as like number 1 in the important properties of enzymes. So each particular enzyme binds only to very specific substrates and catalyze a specific reaction. So we'll see it in the next slide, but a substrate, this is going to be the molecule in a reaction, or the starting molecule, we'll say that, the starting molecule in a reaction. Now, enzymes will bind to substrates. So enzymes will bind to very specific substrates, so they're very highly specific. You can't just throw any substrate at any enzyme and it'll always catalyze that reaction. No, they are very specific and they will catalyze a specific reaction. Our next property of enzymes is that they are extremely efficient. So under really optimal conditions, Enzymes can catalyze reactions at rates that are from 100 million to 10 billion times more rapid than similar reactions occurring without enzymes. So they're so efficient, and they can advance these reactions and speed them up at really crazy rates. So having an enzyme is really important for a lot of reactions. Then our third important property of enzymes is that they are subject to cellular controls. So the rate of the synthesis of enzymes and their concentration of any given time is really under the control of the cell's genes. Enzymes are very highly regulated by the cells that they are within. So here we have this orange globular protein, this enzyme here, and then we have this substrate, this purple colored molecule here. So remember that enzymes lower the activation energy of a chemical reaction, and they really do so by decreasing the randomness of the collisions between molecules. So in normal conditions, you may have two separate molecules here in a solution, and they're bouncing around and bouncing around in the solution, and they just happen to bump into one another, and then this is when they react with one another. And it's really just pure randomness with their movements that they come into contact with one another and react, create bonds, destroy bonds, that kind of thing. Enzymes help to bring these molecules together, and so it's no longer just a random reaction process. These enzymes bring the substrates together and bring them in close proximity to one another so that the reaction can take place. Enzymes also help to bring the substrates together in the proper orientation so that proper reactions can occur. So here at the top here, we have sucrose and water. So the enzymes and the substrates will come together at the active site of the enzyme here, and it forms this enzyme substrate complex. So in this particular reaction, this enzyme sucrase is helping the hydrolysis reaction occur to take sucrose and break it up into glucose and fructose. So this enzyme is going to catalyze the reaction and transform the substrates, which in this case is sucrose, and it transforms it into the products. Now when that reaction is complete, the products leave the active site of the enzyme, and the enzyme, you can see it's unchanged, didn't change the enzyme in any way, and then it's free to go and do the exact same thing over again. So we're going to go back, pick up another sucrose, and catalyze another reaction. So they're very efficient and very specific. You can see this active site in this particular enzyme here is only going to fit this particular molecule. It's going to be a physical barrier right here, a physical specificity, mainly. There's a couple other factors that come into play, but we can just think of it as a lock and key fit. There's a specific key that fits into a specific lock, and no other key is going to fit into that lock. So that's pretty much how enzymes work. Okay, and just quickly a bit about DNA and RNA. Now, nucleic acids, these are named because they were first discovered in the nuclei of cells. These are going to be really huge organic molecules that contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. So nucleic acids. Now, Nucleic acids are of two different varieties. The first one is deoxyribonucleic acid, otherwise known as DNA. That's going to be the first type of nucleic acid. And the other one is ribonucleic acid, otherwise known as RNA. So DNA forms the inherited genetic material inside each human cell. In humans, each gene is a segment of a DNA molecule. Now, our genes determine the traits we inherit, and by controlling protein synthesis, they regulate most of the activities that take place in body cells throughout our entire lives. Ribonucleic acid, on the other hand, this is the second type of nucleic acid. RNA relays instructions from the genes to guide each cell's synthesis of proteins from amino acids. So pretty much here we have this small portion of a DNA molecule. We can see those bonds between the nucleic acids. And each nucleic acid, or each DNA nucleic acid, is going to consist of a phosphate group, a deoxyribose sugar, and a base. And there's 4 types of bases: adenine, guanine, thymine, and cytosine. And they're all bonded together with those hydrogen bonds. So here's a really good table kind of just showing the differences between DNA and RNA. Kind of the main differences being the deoxyribose sugar versus ribose sugar, and two strands in DNA versus only 1 strand in RNA. Lastly, we're just going to talk about adenosine triphosphate, also known as ATP. So this is going to be the energy currency of living systems. ATP transfers the energy liberated in these exergonic reactions to power cellular activities that require energy. Essentially what that means is that when we break bonds in ATP, it releases energy. So we're going to break this bond almost always, and it releases energy. Now that energy can go into a different reaction. For example, the reaction of bonding glucose to fructose. Creating this bond between glucose and fructose requires an energy input. So the energy broken from the bond breaking in ATP will go towards creating that bond. What that means is that ATP provides an exergonic reaction. So here this is exergonic, which means that it releases heat. And this reaction up top here is going to be endergonic. It's going to need energy, energy in the form of heat is often what it is, but it's going to require energy in order to create these bonds. ATP is really used for a lot of things, or the energy derived from ATP is used for a lot of things. It's used to power muscular contractions, it's also used for movement of chromosomes during cell division. It's also used for the movement of structures within cells. It's used to transport substances across cell membranes. And then also what we've already discussed, the synthesis of larger molecules from smaller ones. Essentially building bonds. And that will be the end of chapter 2.